Hello everyone, welcome to Get My Class. Let's solve this question. The question says genetic variations can be introduced in plants by dash where bases in the genes can be altered. Option A says mutation, option B says somatic fusion, option C says tissue culture, option D says somaclonal propagation. Let us understand the question. It says genetic variation can be introduced in plants by dash where bases in the genes can be altered. So first of all, we have to understand what is the meaning of genetic variation. What do you mean by genetic variation? That is nothing but variation in the genetic makeup. That is any kind of change in the genetic makeup. Genetic makeup. Okay, now the genetic makeup, when we say, it can be with respect to the nitrogen base, it can be, it can be like when you try to change the nitrogen base, it can bring about change in the genetic makeup, right? So it can be in one nucleotide. It can be changed in the type of nucleotide which is there. It can be changed in the stretch of the DNA fragment. You can say DNA fragment. Okay. Okay. It can be changed in the chromosome structure. Okay, that can be it can cause structural mutation or structural variation. So all these will bring about genetic variation. Now here in the question, it specifically mentions where bases in the genes can be altered. So we are talking about alteration in the bases. So when they are talking specifically about alteration in bases, that can be done only by mutation. It can be done only by mutation, because mutation, you can mutate the genes either one base or one nucleotide, 100 base, 100 nucleotide. Okay, so you can cause mutation and then you can induce mutation or bring about mutation that will change. Again, mutation can be target based. If you want to just mutate one nucleotide or one nitrogen base, you can do it, and that is possible only with the by the process called as mutation. But if you go for other process like somatic fusion, it does bring about change in the genetic makeup, but it doesn't change individual bases. Okay, so that we have to consider. So let us see. Option A says mutation. Yes, that is the correct option. You can alter the bases here. Option B says somatic fusion. Somatic fusion is the fusion of the somatic protoplast in case of plants, or you can say somatic cells in case of animals, where it is fused and you get a somatic hybrid. So you're not altering the nitrogen base, or you're not altering any base as such. Okay, you are actually mixing up the genes. So that is not alteration of base. Okay, hence it is voted. Option C says tissue culture. Tissue culture may not cause genetic variation. Further, if you continuously keep culturing, that may give rise to variants. Okay, but again, you cannot target a specific base there. Hence, it is ruled out. Option D says somaclonal propagation. Somaclonal, as the name indicates, we are talking about genetically identical. Okay, identical organisms here. But then this happens in case of plants. They do, where they produce somaclonal, somaclonal clones. You can say somaclones, which that means they are genetically identical. But here we are talking about genetic variation. So this is totally ruled. So what is the option? Correct option is option A. That is mutation. So mutation can be utilized so as to alter the basis of nucleotides of a particular gene, and which can bring about genetic variation. Okay. So I hope you understood the explanation. Thank you, everyone.